Think of the cell membrane as a busy border checkpoint. Some travelers naturally drift through because the concentration gradient pulls them downhill. This is passive transport. Active transport, in contrast, is like helping someone climb up a hill. The cell has to invest energy to push molecules where they wouldn't normally go. Active membrane transport is the movement of substances across a cell membrane against their concentration or electrochemical gradient, meaning the cell pushes them uphill rather than allowing them to drift passively. Let's place active transport in its clinical setting because these ion pumps quietly maintain the electrical and chemical stability that the body depends upon. First, consider the nervous system. After a neuron fires an action potential, it needs to rapidly restore its ion gradients so it's ready for the next signal. Sodium potassium ATPase pump is the workhorse here. By pushing sodium back out and potassium back in, it reestablishes the resting membrane potential. Without this reset, neurons fall silent, leading to symptoms like weakness or paralysis when the pump is disrupted. Next, muscle cells rely on the same pump to maintain their resting potential, which sets the stage for controlled contraction. Then shift to the gut and kidneys. Both organs depend heavily on secondary active transport. Your sodium gradients drive the movement of glucose, amino acids, and many electrolytes. So active transport isn't just a molecular detail, it is the foundation that keeps excitable tissues responsive and epithelial tissues absorptive. By the end of this session, you comfortably explain what active transport is, walk through the sodium potassium pump cycle like an examiner, and connect these mechanisms to gastrointestinal, renal, and nervous system physiology. You not only learn the facts, but you'll also be able to apply them in structured exam answers and link pump dysfunction to real patients. The active transport is divided into two types: the primary active transport and the secondary active transport. Let's set the foundation clearly. Primary active transport refers to membrane transport in which the transport protein itself directly uses adenosine triphosphate, we call that ATP from now, to push ions against the electrochemical gradients. Here we see the plasma proteins, transport proteins, and adenosine ATP. The electrochemical gradient here is in this direction for sodium ion because it has a larger concentration extracellularly than intracellularly, and as such, the electrochemical gradient is in this direction, which means to be pushing ions in the other direction against this normal electrochemical gradient that flows in this direction. Potassium, the electrochemical gradient should flow in this direction, but that's intracellular to extracellular due to the higher concentration of potassium ions intracellularly than extracellularly. But what the active transport does is that it pushes the potassium against its electrochemical gradient, that's extracellularly to intracellularly. Now, in other words, adenosine triphosphate ATP supplies energy on the spot, and the pump converts energy into movement. When ATP breaks down into adenosine diphosphate, breaks down, and phosphate ion and energy, the energy released is transferred to the pump often through phosphorylation. This energy drives a conformational shift, forces ions uphill, regardless of how steep the gradient is. That direct coupling is what makes the process primary. You see here, after phosphorylation, phosphate ion, there's a conformational change in the transport protein, changes shape, and attached sodium is pushed against its electrochemical gradient into extracellularly, while potassium attaches and is pushed against its electrochemical gradient intracellularly. Examples include the sodium potassium ATPase that maintains the classic 3 sodium ion out and 2 potassium ions in, resetting membrane potential and preparing excitable tissues for the next event. Another example is the calcium ATPase, which hides calcium inside the sarcoplasmic reticulum, allowing muscle fibers to relax after contraction. Finally, the hydrogen potassium ATPase in the gastric parietal cells that produces the highly acidic stomach lumen by pumping protons out in exchange for potassium. To summarize the mechanism rhythmically for exams, ATP binds, ATP hydrolysis, pump changes shape, ions move uphill. Potassium. A simple diagram of ATP entering and ions being pushed across the membrane captures the entire concept visually. It earns uh, faultless marks. The sodium potassium pump is a primary active pump that directly uses the energy from ATP to move sodium and potassium in opposite directions across the membrane. It's built from two main subunits, alpha and the beta units, alpha beta units, with the alpha subunit handling the work of binding ions and ATP, while the beta subunit steadies the protein within the membrane. As ATP binds and is split into ADP and phosphates, the released energy causes the pump to shift its shape, almost like a rotating gate. That shift opens the pump to the outside, allowing the 3 sodium ions to slip out into the extracellular fluid. In that new outward facing position, the pump encounters potassium and gently catches 2 ions from the outside. When the phosphate group is released, the gate swings back towards the cytosol, returning the protein to its original shape and releasing the potassium inside. 4 steps: ATP, hydrolysis, conformational shape, release of sodium, binding of potassium, release of phosphate, it changes back, sodium and potassium is released intracellularly. One subtle feature of this cycle is that it emits 3 positive charges while taking in only 2. 3 positive charges, taking in only 2. This creates a net outward current, and that is why we call the pump electrogenic. It's a favorite phrase of examiners because it neatly captures how the pump contributes to the resting membrane potential. As the pump runs continuously, it maintains the low intracellular sodium ion concentration that epithelial cells rely on for secondary transport. It keeps intracellular potassium high to stabilize the resting membrane potential in excitable tissues, and it prevents cells from swelling by limiting osmotic water entry. Secondary active transport refers to a mechanism in which a solid is carried uphill or downhill using the potential energy stored in another ion's gradient, most commonly the sodium gradient. What makes it secondary is that the transporter itself never comes into contact with ATP. Instead, it relies on the gradient generated earlier by the sodium potassium ATPase, which hydrolyzes ATP and establishes the conditions these carriers later exploit. Here you can see the secondary active transporter and the primary active transporter. The primary active transporter, in this case sodium potassium ATPase, is the one in contact with the ATP, while these never come in contact with ATP. But you use the energy of ions that have accumulated extracellularly, in this case sodium. You can imagine the sodium gradient as a stress spring packed with stored energy because sodium has been pumped outward and now wants to rush back in. Secondary transporters attach another molecule to sodium's inward pull. As sodium moves downhill, the paired solute is lifted uphill or downhill, in this case, downhill, in a beautifully economical exchange of energies. If the transporter moves sodium and another molecule in the same direction, like this, in this case, call it a symporter. Classic example is the sodium glucose transporter seen here, cotransporter in the intestine, which allows glucose to enter the cell by hitching a ride on inwardly directed sodium. If sodium enters but another substance is moved outward, the carrier acts as an antiporter. The sodium calcium exchanger in cardiac muscle demonstrates this perfectly. But the key message here for exams is that secondary transport absolutely requires energy, but it obtains it indirectly. To appreciate symport and antiport mechanisms, it helps to picture the membrane as a busy two-way gate where ions and solutes move in coordinated patterns rather than random flows. Symporter carries two different distances across the membrane, the same direction. Different in the same direction. Sodium glucose transporter offers a familiar example. Sodium moves down its gradient into the cell and glucose is drawn in alongside it. This is how the intestine and kidney reclaim glucose efficiently using sodium's um, downhill gradient to pull glucose back up. Antiporter, by contrast, works like a controlled exchange. One molecule enters while the other exits. The sodium hydrogen exchanger is a classic case, using sodium's inward drive to push hydrogen ions out, quietly helping cells regulate their internal pH. In the cardiac muscle, the sodium calcium exchanger uses the same principle but applies it to calcium. Uh, Sodium enters as calcium is moved out, allowing the cell to lower its calcium concentration after each contraction. This antiport exchange is one of the key steps that lets the heart relax smoothly between beats. Active transport, especially the sodium potassium ATPase, is essential for maintaining the resting membrane potential, the steady voltage that allows nerves to fire and muscles to contract. The same sodium gradient also powers nutrient absorption as sodium coupled carriers draw glucose and amino acids into intestinal and renal epithelial cells. By continually exporting sodium, the pump also supports cellular volume regulation, preventing osmotic swelling and ensuring long-term cell survival. Clinically, these mechanisms appear in familiar settings. Digoxin enhances cardiac contraction by inhibiting the sodium potassium ATPase. Diarrheal dehydration is treated using sodium glucose symport, and hypokalemia disrupts membrane potentials, leading to weakness and arrhythmias. Altogether, the sodium gradient and the transporters that maintain and use it lie at the heart of both normal physiology and common clinical problems, influencing every heartbeat, neuronal signal, moment of nutrient uptake. Now, importantly, when exam questions ask you to describe a mechanism or explain a physiological process, the examiner isn't just testing recall, they're looking for a logical, structured story. So let's build that story piece by piece. First, always open with a clean definition. This places the topic in its physiological context. If relevant, add a brief classification. For example, primary vs secondary active transport. Next, include a clear, labeled, and proportional diagram. Even a simple pump, channel, or gradient sketch can secure marks that paragraphs sometimes miss. Then move into your stepwise mechanism. Walk through the events in sequence. Ion movement, voltage change, conformational shift, functional outcome. This cause and effect rhythm helps the examiner follow your thinking and immediately elevates your answer. After that, add a short section on functions, that is, what the mechanism achieves for the cell or system. This includes maintaining gradients, enabling absorption, and stabilizing membrane potentials. Now, bring in clinical correlations. You can mention a disorder, consequence, or drug effect that arises when the mechanism is impaired. Finally, wrap up with a summary table, perhaps comparing steps, ion rules, or differences between pathways. This reinforces your answer and leaves a clear final impression. The overall strategy is simple: define, draw, explain, apply, then summarize. Quickly, the key takeaways from today's tutorial is that primary active transport relies directly on the energy of ATP, while secondary active transport cleverly uses the sodium gradient created earlier by the sodium potassium pump. The pump itself is electrogenic because its unequal exchange of sodium and potassium creates a small but meaningful outward current that shapes the resting membrane potential. Symporters move two substances in the same direction, often facilitating the absorption of nutrients like glucose. Antiporters move substances in opposite directions, playing roles in pH control and calcium excretion. Together, these transporters form a coordinated system that keeps electrical signals sharp, nutrient uptake efficient, and ionic balance stable. Quick quiz at end here reinforces the essentials: the electrogenic nature of the pump, classic symporter example, such as the sodium glucose carrier, and the rise in intracellular calcium that occurs when the sodium calcium exchanger is inhibited. These questions capture the heart of the topic. That is how ion gradients create the energy landscape that cells depend on, and how transporters translate that energy into function. Well, this brings us to the end of today's session. Thank you for joining this MedPhysio tutorial. Keep exploring physiology with curiosity, and remember, every gradient tells a story. See you in the next lesson.